What I'd like to go over is what we've been looking at for rapid detection technologies, really targeting the food industry, and there's a lot of motivation for this. If we look at what companies are spending on hold and release, of course, information faster is always better and cheaper. If we look at food safety, some of the laws that have been proposed, of course, with FSMA saying that agricultural water needs to be tested weekly. Right, so if a farm, if we think of a farm that has to test agricultural water for E. coli and comes back with high E. coli counts days after they've tested it, right, because they're sending the samples out, then they're told not to use that water that they used days ago. So it doesn't really make sense, and so what a lot of things are pointing to is how can we really detect things a lot faster, at least as far as a screening process than we do now. So what I work on is biosensors, and I want to show you an example of one of the first documented biosensors. This is a person who's diagnosing diabetes. We've gotten well beyond tasting urine at this point, thank God, but that's how they used to diagnose diabetes back in 1670. Certainly technology has helped a bit. And I want to show you the first disposable biosensor. So this is what they used, of course, in the coal mine to see if the oxygen levels were sufficient. In this case, a biosensor, it basically means we're using something biological to detect something. It doesn't mean necessarily that we're detecting something biological, but just that we're using biology to do our detection, whether it's antibodies, nucleic acids, or receptors. Again, poor retirement plan for the canary in this case. So we're looking at conventional versus rapid methods. That's where we started. So of course, conventional methods being our traditional methods, what we use with plating, like you can see up here. And this is a microfluidic device where we're trying to shorten things, detection times. One of the things we've realized is, this works great if you look at sampling blood or something like that. But when you get to food, people usually run when it comes to the detection area because you cannot fit a 25-gram sample in this inlet here. It becomes very difficult. And the missing part of the research is and has always been sample preparation. So it's one of the things we've been looking at. Oh. Oops. So we need better biorecognition. So biorecognition means what am I going to use to know that there is some kind of, say, pathogen in this food source? What am I going to use to recognize it? Now, typically you can use antibodies or something like that. We're trying to see how can we move beyond that and look at other sources. This is, of course, science fiction, but this is a nanorobot, which we've seen in different magazines trying to portray what nanotechnology could be. I don't think we're ever really going to get to that. We're making these little nanorobots, but if we think about what we do have the technology for, resembles something like this. And for those of you who might know, this is a virus, right, on top of a cell. You can see the cell here and the virus is up here. They resemble nanorobots, right? And as we have seen recently, they're very programmable. We can have them do things that we want them to do. We can't really target them too well, so we've been working on that. Here's an image here of T7, just another bacteriophage, which targets E. coli. So what we come up with as a plan to do rapid detection, we took the bacteriophage here, and we genetically modified it so we can conjugate it directly to a magnetic nanoparticle. So that is a little magnet covered with glass, which we bind to the outside of the virus. Right? Now we can actually move that virus around in a sample, a liquid sample. It can bind onto this bacterium here. Of course, it'll lyse eventually, releasing a lot more bacteriophage. And then we can either, using some kind of device down here, count the increase in bacteriophage, or we can have that bacteriophage force expression of, say, a peptide or something else, which we can count later. Enzymes have been used quite a bit recently, something like luciferase. First thing we wanted to do was work on this magnetic nanoparticle. So if you guys remember, if you've taken food engineering, remember Stokes' law and why we homogenize milk, right, to make smaller particles move slower, right? In this case, we have a nanoparticle. They move really slow because they're extremely small. We had to take care of that problem because separation obviously would take days if we put all these things in a sample, food sample, and tried to separate out all the bacteria. So we had to come up with a special nanoparticle. So this is a cobalt-containing nanoparticle, which we've been working on, where we can see here, this is what iron oxide, the traditional iron oxide nanoparticles would look like. And here's our cobalt-containing nanoparticles. In the time that this would normally take, this would take days to separate. And our video here, we can see the cobalt-containing ones separate really quick. So if we were to throw these into a liquid sample, we can yank them out really fast. Now the theory, of course, if these have bacteriophage on them, and they bind onto a particular bacterium, it would be yanking that out of solution, at the same time infecting it, which we would use for our amplification and detection. Yep. So this is what we have. We modified it, so we have our bacteriophage. We had these magnetic nanoparticles here. This is my cartoon because it's prettier than the actual thing, but we can see the silica shell and the magnetic core. And these things separate out and grab bacteria pretty well. So we did a study where we took bacteriophage on nanoparticles, bacteriophage on micron scale particles, and compared them to antibodies on the two types of particles as well. Here we can see this is bacteriophage on micron scale particles, which have pulled out bacteria. And this is the nanoparticles with the bacteriophage, which we've separated bacteria and taken a TEM of them as well. So they're able to pull them up quite well. The results we can see here, so phage on, this is phage on microscale, nanoscale, and then antibodies on nanoscale and microscale in the order. So in this case, we were looking at E. coli because we use T7. Now typically, we're more interested in the food industry, and the lower end here, there's a concentration of bacteria here. We're interested in the lower end here because obviously when we get really high, then things are pretty obvious that there's an issue. So when we look at this area here, we can see that the bacteriophage were much more efficient in pulling out the organism than the antibodies were. And I should mention that the bacteriophage in this study, we did in half the time. This is because antibodies took half an hour to bind. If we let the phage take that long, they would start to lyse the organism, and that would cause a problem with separation. So in this case, we were able to separate them out much faster and much higher capture efficiency. Now, capture efficiency I'm talking about, if we put 10 to the 2 organisms in a sample, put the solution or the particles in with the phage, this is the percent that we're able to recover. So a much, much higher capture efficiency. Then we went on to detection. Well, if we then detect the phage that came out of the replication of that, right, because the ones that we started with are attached to magnets, so we can differentiate those, we can measure the phage that came from the infection. If we can measure that quantity of phage, we can kind of backtrack and see how many E. coli did we have initially, right? Because remember, this is only going to infect a viable cell. So this is also telling us if we've got cell viability in this case. Unlike antibodies, which very often will tell us total cell count, viable and non-viable. We looked at using fluorescent nanoparticles. These are particles we also made in our lab. So this would be a traditional lateral flow assay. So this is like a pregnancy test, where we put the sample on, it runs up, we get this line here, which is actually gold nanoparticles, if it's positive, and only one line if it's negative. And of course, if we run a fluorescent, we get about 3 orders of magnitude better sensitivity and lower limit of detection than we got with the gold nanoparticles. So in this case, we're able to detect about 10 to the 3 bacteriophage. Now, if you consider that when bacteriophage infects an E. coli, you get about 10 to the 2 bacteriophage out of there. Right, so this means that our detection limit for E. coli is actually extremely low. We can do this electrochemically as well. So we use an electrochemical sensor, which we developed here. This is not the price of the sensor. This is just to give you an idea. But we hope to make it somewhere around that price, but that's not going to happen. So we developed a small electrochemical sensor to be able to detect the increase in phage. And we want to do this because we need something that's fairly simple and fairly robust. Using the lateral flow assays, of course, have the limitations with sensitivity. So we put this thing together, and we can see here we're able to distinguish 3 times 10 to the 3 bacteriophage. Now this, of course, I have to preface this, we haven't actually done the E. coli counts, but if we were to go with what our studies have shown us, which we get a burst size of 10 to the 2, that would be a very low concentration of E. coli that we can detect. Right? So this isn't real data, this is just, that one's mocked up. So the question is, that's great, right, for solid foods, but what can you, or for liquid foods, but what can you do for solid foods? Because obviously, a lot of what we see in the food industry and what we have problems with, say this is spinach and this cookie dough, magnetic separation doesn't work too well for those because you can put your magnets in and try to pull them out, and of course you're stuck in the cookie dough, so it's not going to work too well. So what we wanted to see is what can we do as a sample prep for this type of food. The other issue that we know we've encountered, this is a paper that came out I think 2011, showing this is E. coli inside a plant. Right? So we know they integrate inside the plant and they internalize. Right? So these are very difficult to get out once they're in. This is a paper by Solana, not my group. But so we see all these organisms in here, and that's the big problem. How can we get those out to be able to detect them? Right? Because they're inside of the plant and very difficult to get out. And so what we come up with, if we imagine this is the bacteria inside of the plant, introducing enzymes. Right? So if we do an enzymatic treatment where we can slowly degrade the plant matter from, one too many, from around the organism, we can release them into a liquid. Right? So we then are able to detect these or grab them or pull them out once this has been degraded. Right? And we look at the results of this. We did this with cookie dough. And cookie dough, of course, we're using amylase, and we're using beta-glucanase and lipase. Right? So these are cocktails that we put together and optimize. And we can see here we start with the control, where we have 0 minutes, and then we go to 40 minutes, and we've got a complete liquid. Right? So this cookie dough has been turned into a complete liquid, probably very delicious, but it's now a drink versus a snack. Right? On the bottom, we can see the spinach, where we go in 3 hours. I should mention that this is very liquid up here. There's still some of the fibrous material up here. This, of course, we're using pectinases. We're using all sorts of everything meant for a plant. Now, the enzymes that we've gotten out of this, and we're working with Novozyme on this, has really come from biofuel research, right, where you're looking to break down the plants. In our case, we found that using these enzymes here, we get about 4 times more of the capture efficiency or extraction of the bacteria than we get when we don't use the enzymes, because we're completely breaking it apart. I should mention, this is, if we filter this, we found that we got a 40% loss in mass. Are we on time? Good. So we pass this through a filter, and we can see a lot of it has just been solubilized or as in particles small enough to pass through a 10-micron filter. Another area we've been getting into is to detect things faster. What if we don't even have to really do a sample prep? Right, so we've got a nose here, which is how we do all our volatile testing every day, right, when we sniff our coffee or our food. And this is what a lot of consumers are interested in, right, because they are the ones sniffing the coffee or whatever food product. And we want to make sure that our food smells good when they open it. Right, so we see this obviously with wine. It would be very beneficial if we could do very efficient volatile testing using wine. This was for you, Brent. I put in some other liquids. And soups, other foods like this. But then we look at this from a quality standpoint, right? So what if we could detect mold, right? Things like this, or fungus, or anything that might be a bad quality attribute, or even a food safety attribute. If we could smell these things in more of a bioelectronic nodes, it'd be great. So if we started putting together a sensor where we can kind of mimic what a dog can do. So when I was coming back from Germany, I think 3 years ago, my mom, who lives in Germany, had given my wife a banana for the plane, forced it on her, right? Take a banana. And she made it into the airport, and of course a dog came running over to her, sat down in front of her, and one of these people here came over and said, "Give me your bag," took out the banana. And that just amazed me, because this dog came from on the other side of the room, right? So she got in trouble, which is fine. Not a lot of trouble. But so it made me think, like, this is amazing. We don't have an instrument that can do that. There's nothing we can manufacture that can smell a banana from the other side of the room, right? And so I wanted to make one. Working with the company now and trying to develop something, and we look at how do smells, how do we sense smells, right? We use receptors that are in our nose, right? Which are usually G protein coupled receptors, which can bind onto the volatile. Now there's a lot of research on how that binding is actually happening, but we know it can recognize certain smells, right? And when this happens, it sets off this little reaction where things happen down here in the bottom. But the main part is we have a binding effect up here, right? Something is happening. Some kind of biorecognition is happening. What we found, and if we take 2 electrodes here and put these are conductive nanotubes connecting the 2 electrodes. Now, I should say my research has evolved faster than my drawings have, because now we just have one tube going across continuously like this. But if we put the receptors on these tubes, we functionalize it with the receptors. What we find is if that volatile is near, as we can see in this case, these receptors will start to bind to it and change the conductivity of what's underneath it. Right? So now we can actually measure this binding effect. So we're using conductive nanowires now to do this. And what we found is, I should explain this, we use hexanal, which is, as some of you know, a byproduct of lipid oxidation. Right? So if we inject hexanal across this, and then air, right? So this is just ambient air. Hexanal air, we can see this thing is able to detect hexanal quite well. Right? This is still fairly early in our research progress, but this is showing the kind of technologies we're able to find. Let's use the olfactory receptors, say, from a dog, rat, and mouse, and a human. Now, these are all synthesized. I'm not yanking them out of the animal. But we're able to do this and smell these particular smells. This particular receptor that we are using was known to react with the hexanal group. So we also have some that are known to react with volatiles from mold and from fungus. So one of my pet peeves this is switching gears. One of my pet peeves when I look at detection is the microfluidic lab on a chip, right? Because most of the people who come up with a lab on a chip are really developing what's called a chip in a lab, right? It's got all these pumps and everything on it. It's nothing that's portable. How can we make these things portable? So one of the things we said is, let's not put pumps in it. Let's find a way to do fluid transport so we can do all these little miniaturized reactions on this chip without using any pumps, right? So just use capillary flow. And we know capillary flow is controlled by surface tension, viscosity, and contact angle, which means how hydrophobic is that plastic that we're in. So we said we can control contact angle. We can control how hydrophobic something is, right? It's something called electrowetting. Now you can see here we have a hydrophobic layer on an electrode. And so this is a water drop. This is an electrode going in, and it's sitting on an electrode. And you can imagine if you've ever put Rain-X on your car, the more hydrophobic something is, the more the water bubbles up on it, right? So if we were to adjust that, make it hydrophilic on demand, we can make something go from hydrophobic to hydrophilic. And we put this inside some kind of device, and we can essentially create a valve. So what we wanted to see is, can we just stop fluid flow by, this is a regular electrode, unmodified, modified electrode, have it stop when it goes through a channel so we can get rid of all these pumps. And we saw here that we could. This is a mock-up where it did stop at the electrode and then continued once we added a small voltage, about 2 to 4 volts. And so here we'll see my graduate student's shaky hand putting 3 colored devices into these circles here. There's a microfluidic channel where the red is. This up here is just an absorbent pad, so all the colors are going to go up there, the different colors. So we can see you're putting in blue, stops at the second electrode it meets, yellow, stop it at the second electrode, and then a red one, well, it turns red, but I won't go into that, stops at the second electrode. Now we can release these at different times, so if we think about, if we can now do all this controlled release, we can do, say, an ELISA reaction right inside a chamber like this. So she releases blue first, then yellow, and then red. Now this is an expensive way to make a tie-dye pattern, but it's in my field it certainly is going to be somewhat helpful, more to me. So with that, I gave you an overview of the things we're looking at and the directions we're going to try to make it so we can detect things much faster in the food industry. I really emphasize that the thing that's missing in general is sample prep, and to be able to go from, say, 25 grams of something into 10 microliters. And for those of you who have been at the trade shows, have been, say, on the floor at IAFP, there are hundreds of people who say, I've got this great device that can detect a single bacteria. Just put your clean 25 microliter sample in here, and I'll let you know if there's any listeria in there. But the food industry doesn't work that way as easily. So it's the need for research and sample prep is where I really think we need to go. Okay. Thank you.